involves your skeletal muscles. Okay, autonomic portion of the peripheral system includes sympathetic, which is arousing, and parasympathetic, calming. So I showed you a nerve cell earlier, which includes the cell body, and that's the biggest part of the neuron in diameter. It contains a nucleus like all eukaryotic cells do, except for red blood cells. At least when red blood cells are mature, to make room for hemoglobin. But the nucleus, of course, can do transcription and replication. But if you do transcription and make messenger RNA, of course, we can make proteins, which include neurotransmitters, which are special types of hormones made by nerve cells. The dendrites, as I pointed out earlier, receive information from sensory receptors, and the axons allow those signals from the dendrites, the cell body, to travel from the nerve cell to the axon, and that will help release the neurotransmitters to the recipient cell. But this axon is covered by myelin sheath, which I mentioned earlier. But what's special about it is made up of special lipids called sphingolipids. In previous chapters, we discussed which lipids were in a membrane in other cells besides nerve cells, and those included glycerol phospholipids. Sphingolipids are similar to glycerol phospholipids in that they have two hydrophobic fatty acid tails, which are shown on the left. One is labeled as sphingosine, and one says fatty acid, but they're both fatty acids. So actually, the whole structure in orange and blue is sphingosine. Where it says R, that's where you're going to have a modification from the sphingosine. So it's like sphingosine itself, which is in red and blue, is the building block, like glycerol is for other lipids. And the RNA, and then it has a little text box there. It says R can be a ceramide, okay, or a ceramide plus, or it can be a sphingomyelin, or a glycosphingolipid, which we'll discuss. Possibly. But this is to help for insulation, just like you have insulation in your home. Sometimes they call these long axons a nerve fiber. Okay. So here's this picture again. Again, dendrites receive information from another cell, or they might be the first nerve cell to receive a signal, just like touching the hot stove. And then again, that signal has to be identified, and then based on that signal, the cell body, in particular the nucleus, will act on that and make a neurotransmitter, and that will travel along the axon where it's insulated by the myelin sheath and reach the synapse, where there will be a small gap between it and the recipient cell. Okay, and there's a note there that it's like any other eukaryotic cell, and that it has all those organelles that we're used to seeing in animal cells. There are various types of neurons. There are motor neurons. They accept nerve impulses from the central nervous system, like your arms, your legs, your feet, your hands, and transmit them to muscles or glands. Sensory neurons accept impulses from sensory receptors. Okay, those receptors may be activated by hormones, and then they transmit them to the central nervous system. Okay, interneurons convey nerve impulses between various parts of the central nervous system. There's various types of neurons. There's a unipolar neuron. Okay, that's just one way. Bipolar neuron, it's two directions. You have pseudo-unipolar neuron. So a unipolar is usually one direction, but this one does look like the unipolar one. However, it does branch off into two different sets of dendrites there. Multipolar neuron is the most common because that you can see you have dendrites, a cell body, an axon, and the synapses. So the most common one's the multipolar neuron. You have glial cells, okay, which can provide support to the actual neuron, and they show a multipolar neuron here. So they support, protect, and nourish the neurons. There's more of these support cells than neurons, 10-to-1 ratio in the brain. They fulfill many functions. And the brain tumors are usually caused by mutations in the glial cells. They are showing you a pseudo unipolar neuron over here, where we see it has two different directions for the axon. But that would be helpful for the peripheral nervous system. Again, there's your arms, legs, for example. In the glial cells, which are offering support and nourishment to the actual neurons, in the central nervous system, there's various cells. They're important. There's different types of glial cells. There's oligodendrocytes. They form the myelin sheath, which acts as insulation. You have the astrocytes. And as their name suggests, let's see if there's a picture, they kind of look like stars or a big sun. They provide nutrients and structural support. Then you have the epidermal cells. They produce cerebrospinal fluid that cushions the neurons. And we have the microglia, scavenge pathogens and dead cells. Yeah, that's important. We don't want a brain infection. The peripheral nervous system includes Schwann cells, which form the myelin sheath there. So that's interesting. It's different cell types that make these sheaths depending on if you're in the central nervous system or peripheral system. Satellite cells, they also provide nutrients and structural support to neuron. So they have the same function as astrocytes. Maybe think of satellites are in outer space, and astrocytes sound like stars. So that's why they have similar functions. So this is showing a reflex arc when you respond to a stimulus, and how the central nervous system knows about it after the peripheral system has told them. Okay, so here it's showing you somehow you got a pin in your skin. Okay, and so that's relayed over here that you unfortunately have a pin in your skin. That's relayed along the peripheral nervous system to the central nervous system, where in purple they point out those dendrites. And then it goes back, and those dendrites of that nerve cell create a neurotransmitter to tell through the axons the next nerve cell through their dendrites to do something. And so we see it's traveling through the ventral root there to the muscles of the arm, and having them move their hand and hopefully take the pin out of their arm, and hopefully much faster than I just said that. Now, because nerve cells make hormones in their cell body, it's necessary to go over the cell signaling pathways on how hormones cause effects. There's two such pathways. There's the G protein coupled response pathway and the receptor tyrosine kinase pathway. Both involve hormones binding receptors that they've indicated in green. The GPCR pathway's receptor looks like a snake crossing the membrane many times. That's in yellow. And the RTK receptor to receive this hormone is in two parts, like a salt and pepper shaker, and it actually dimerizes or gets closer together into two parts, which they add phosphate groups to itself upon a hormone binding in the RTK pathway. In the GPCR pathway, a G protein gets activated when a hormone binds that receptor that looks like a snake, and it activates an effector enzyme, which makes a secondary messenger, which is another small molecule. It binds a special kinase that's specific to that secondary messenger. Okay. And then as a result, that kinase adds as a phosphate to some protein after it takes it from ATP. Similarly, the RTK pathway, once the receptor is phosphorylated, that information is conveyed indirectly by an adapter to RAS, And RAS activates a kinase called MAP kinase, which will also phosphorylate some molecule or protein that will eventually lead to a cellular response. So they do it slightly differently, but in the end, they activate some protein or change the protein's response by phosphorylating it. Neurons communicate with each other and muscle cells, and these signals are possible because each neuron has a charged cellular membrane. Earlier in the semester, we talked about the sodium-potassium pump, which is shown here, which always has 3 sodiums go out as 2 potassiums go inside the cell. So as a result, since you have more sodium on the outside of the cell versus the similarly charged potassium on the inside of the outside of the cell usually has a positive charge and the inside of the cell has a negative charge. Okay. Now this charge gradient where it's more positive on the outside versus the inside does change when the neurons get active during an action potential. So if we, so we're looking at charged membranes here. We're looking in teal at the light blue or teal protein in the membrane, and we see that it's closed. Okay, and there's these circular objects that want to come in. Well, they can't come in when it's closed, but it can open in response to a nerve impulse. And this may be when sodium enters the cell. So if sodium enters the cell, that changes the gradient there. So I should go back. So voltage-gated sodium channels are activated when there's a nerve impulse. So they're going to be open when there's a nerve impulse. In this example, there was a nerve impulse, which caused the charge across the membrane to change from plus on the outside to negative on the inside. It changed, and then that allowed, in this case, sodium to enter. Okay, and then after that occurred, then it closes so it doesn't allow any more sodium to come in. So voltage-gated ion channels regulate the relative concentrations of different ions in and outside of the cell. So voltage-gated is involved with charges across the membrane. We'll see how these are different from ligand-gated channels. And the difference in the total charge across the membrane between the inside and outside of the cell is called the membrane potential. Resting neurons, they have a resting membrane potential or difference in charge across the membrane of usually minus 70 millivolts. Okay, so it's that much more negative in the inside than the outside. Now that's not always the exact number. So this resting membrane potential is created and maintained by increasing the concentration of cations outside the cell compared to the inside of the cell, so that's why it could be negative. And remember, it's 3 sodium outside to 2 potassium on the inside. Okay. So an action potential is when this nerve cell is going to get away from that resting potential of minus 70 millivolts approximately. So some stimulus from a sensory cell or another neuron will depolarize the target neuron to its threshold potential. So it's going to depolarize it, so we're not going to have that minus 70 millivolts maintained by the sodium-potassium pump. So separate sodium channels, separate from the sodium-potassium channels, are going to open, and that's going to allow positive ions to enter the cell. So the sodium ions are going to enter the cell, and when the sodium channels are open, that's going to cause the inside of the cell to become positive before it was negative. And these are all or nothing signals. Okay. So once the threshold potential is reached, which is a positive number, then the neuron is going to be completely depolarized, meaning it's no longer negative on the inside and positive on the outside. It's flipped the opposite way, where it's positive on the inside. But once this depolarization is over, we don't want to continually feel pain. The cell must now reset back to the voltage it was before, like minus 70 millivolts, for example. So here we see a neuron or a nerve cell here again with the dendrites on the left, the cell body, which now they wrote soma, which is the same thing, the long axon and the axon terminal, which will end up in synapses. And what they're showing you in response to a signal, the soma end of the axon, where the dendrites are, becomes depolarized. Okay, so now we see all these changes here. Okay, and then so in green is depolarized, but in pink it has not depolarized yet. But eventually that's going to radiate towards the axon. So this charge gradient, where it was positive on the outside and negative on the inside, is going to change and cause neighboring portions of the axon to feel the same effects. So initially it gets depolarized on the far left of the axon, and then as we go, it's going to go towards the far right, toward the axon terminal. They'll get depolarized. But as more and more to the right gets depolarized of that axon, then previous areas of the axon to the left near the dendrites will repolarize or rest and go back to the resting potential so that the cell can be responsive again. But as long as it's depolarized or repolarized, it cannot respond to a signal in that area. So as mentioned in B, the depolarization, where the charge has flipped across the membrane, spreads down the axon. Meanwhile, the first part of the membrane repolarizes. And why? And then because the sodium channels are inactivated and additional potassium channels have opened, the membrane cannot depolarize again. Okay, so once it's, and that's to make sure we're not continually feeling that sensation. So here's an action potential graph where we, on the y-axis is voltage in millivolts over time in milliseconds on the x-axis. And if we look at 1 here, or actually if you look at 5 first, and in the beginning of 1 there, it's at minus 70 millivolts, and then something's occurred, and as long as it gets above the minus 55 millivolt threshold, you're going to have an action potential where now the sodium potassium pump is not operating and causing this depolarization, but sodium ions are rushing in, so that negative minus 70 that we had previously inside the cell, we're not going to have that. So much sodium goes in that we've gone from minus 70 to plus 40. Okay. But we can't keep it like that, because if that was causing pain, it's going to cause more pain. So we have to repolarize the membrane, but it usually overshoots the repolarization. Okay, and now potassium's going out so that the cell will become more negative again, but it overshoots it to more negative than negative 70. It didn't like label it, but eventually then the sodium potassium pump will work, and then we'll get back to the resting state. Okay, and this will occur, this action potential will keep going all the way from the beginning of the axon closest to the cell body to the end of the axon furthest away from the cell body near the synapses. Now, so that was a voltage-gated synapse. A chemical synapse can lead to depolarization, and this causes voltage-gated calcium channels to open. So by the time we get to the synapse or near the synapse, that's going to open up these voltage-gated calcium channels. They only open once they notice that the membrane potential has changed from minus 70 to plus 40, for example. And then these calcium ions, now that are allowed to enter, initiate a signaling cascade that causes synaptic vesicles containing neurotransmitter molecules to fuse with presynaptic membrane. What is a presynaptic membrane? So let's go back to this picture here. So this picture here, where there's the axon terminal, these would be presynaptic membranes. They're before the responding cell. Postsynaptic membranes are beyond the synaptic cleft, the gap between two neurons, or a neuron and a muscle cell. Okay. And this fusion of these vesicles containing hormones here will allow release of the hormones into this little gap between the initial neuron and the responding neuron or the responding muscle cell. So once neurotransmission has occurred, the neurotransmitter must be removed from the synaptic cleft, because otherwise, just like the action potential, we don't want it always on. We don't want this neurotransmitter causing these effects over and over again. Otherwise, the nerve cell or the muscle cell can't respond to new stimuli. So it has to be reset. When it says postsynaptic membrane, that would be after this axon terminal, after the gap, and the recipient cell is responding. Okay. So if we look at a closer look at the chemical synapse at the end of the axon, or the axon terminus, and they've shown that they've enlarged it here. We see in those vesicles hormones in there, the synaptic vesicles, and they're usually from the Golgi body, sometimes in other cells. And calcium helps the release of these vesicles. And we can see once those vesicles fuse with the membrane, the calcium is free now, not the calcium, but the hormones are free now to bind to, in step 4, these receptors specific for these neurotransmitters. And the synaptic neuron, or it could be a muscle cell, but here it's a neuron, is going to respond to that neurotransmitter and either follow the GPCR pathway, which has been a while since I mentioned it, the GPCR pathway, or the RTK pathway, which eventually will lead to something at the very end being phosphorylated that will cause a cell action. Okay. And what we show here is on the left, we see the neurotransmitter is bound to an ion channel. So if the neurotransmitter binds to an ion channel, then this is going to allow certain ions in or out. Okay. But after a period of time, in the second box on the right, we see that these neurotransmitters are degraded or diffusing away, so we can stop this transporter on the second cell from responding, so that it can respond later. A voltage-gated channel instead of a neurotransmitter. So this one here is a ligand-gated channel where the neurotransmitter bound to the receptor, which happened to be an ion channel to let them in. A voltage-gated channel instead is going to sense the changes in the charges across the membrane. So on the far left is a deactivated or closed ion channel or voltage-gated channel. It's closed, where it's positive on the outside and negative on the inside, because there's more sodium on the outside than potassium on the inside. And then once it changes and we see it's flipped, the charge is more negative on the outside of the cell than the inside. Then it opens, the ion comes in, and then it closes. Okay. So that's a little bit different there between the two. So ligand gated, the neurotransmitter physically binds there and opens it like a key in a lock. Here, it's a little bit more abstract. There's a change in the charge, which we mentioned before, how that's mediated with sodium ions coming in, okay, and then repolarizations with potassium going out, which is the opposite direction that the sodium potassium works. Okay, so here's more of it. And then eventually, again, in this case, the neurotransmitter had to be degraded to stop it. And then here, eventually, to stop this, we showed again that the potassium ions go out. Okay. So taking a closer look at cell signaling, and this is specifically for the GPCR cell signaling pathway. Once that pathway is activated to make that secondary messenger, in this case, phospholipase C, degrades lipids in the membrane so that we get diacylglycerol, which is glycerol with two fatty acids still in the membrane, but it frees this polar group called IP3, which is a sugar called inositol triphosphate. That inositol triphosphate is a secondary messenger which will help calcium ions come out of a endoplasmic reticulum. And then that calcium can bind to a kinase. And then once it gets anchored by the glycerol containing two fatty acids or diacylglycerol, that kinase will only phosphorylate or add phosphates from ATP to compounds near it. Okay. And that will change what the cell is doing. Now, there is some good YouTube videos out there, and I have them here. I would definitely look them over before exam next week. How neurons communicate, the action potential, and the synapse. He's a very dynamic speaker. I don't know if I've had you listen to him previously. Thanks for listening for that part there. Now, we're going to focus briefly on the brain, but we're not going to go in extreme detail. Now, if you have lab, we don't cover the brain anymore in lab. We used to, but I think they decided it was too much. The cerebrum, as shown here with the two hemispheres, is the largest portion of the brain in humans, and it's involved in communication with and coordinating the activities of the other parts of the brain. We see this, looks like a division there. It's called the longitudinal fissure, and that gives us our left and right cerebral hemispheres. This is a cross section of a brain. Okay, and we do see the cerebrum there. It's above the corpus callosum. You don't have to memorize all these parts. And we do see the brainstem, which is like leaving the brain as well. The cerebellum is like the next biggest part of the brain almost. And then there's other glands in here. Okay, we're not, there's a lot in here. You could spend a long time on this. But I like this picture better because it does give you some information about what parts the cerebrum controls. If we look here, that they've highlighted them in different colors. Okay, so we see it's sort of in the middle of the brain in the cross section that controls the lung and, sorry, I didn't mean to say lung, leg and trunk and arm and hand and face and tongue. It's pretty much in the middle there is where a lot is going on there. In terms of movement of the body, okay. The visual area, we can see it's over here in the occipital lobe. Okay. And then they have the parietal lobe, which has the primary somatosensory area, including taste. Okay, actually, not taste. And then we have the temporal lobe over on the far left, which involves auditory association area. Okay. Primary auditory and sensory. So you have hearing and speech near each other. And the frontal lobe includes some motor areas, which does involve some movement, which we've mentioned already, where they already had labeled where the central sulcus was, the leg and trunk and arm and hand and face and tongue movement. So the cerebellum, again, we saw, and it's a little hard here with me using my iPad, but the cerebellum is in the lower right here. Okay. And receives it now. It's one that receives sensory input from the eyes, ears, joints, and muscles. And then based on that, we'll send motor impulse out of the brain to the skeletal muscles. It's also involved in balance and coordination. The brain stem, which we see is long. Okay, it's in this picture to the left of the cerebellum. can be made up of the main midbrain, pons, and medulla oblongata. The midbrain acts as a release station for tracking passing between, as a release station for tracks passing between the cerebrum, spinal cord, and cerebellum. Okay, so it's supposed to receive information and relay it to other areas to respond to it. The pons regulate breathing and head movements. Medulla oblongata, you can think of the word lungs in there, contains reflex center for vomiting, coughing, sneezing, hiccuping, which is not my favorite, and swallowing. Okay, so, and that's that long stem going down. Now we're going back to the peripheral nervous system, okay, which it controls things radiating away from your central nervous system, which was in the middle. You can have the somatic system, which is under your control, contains cranial nerves and spinal nerves, gathers information from sensors and conducts decisions to effectors, controls skeletal muscles, which you can control, and you're conscious of this activity. Autonomic system, you're not under, you don't consciously control these. Controls smooth muscles, cardiac muscles, which I'm relieved I don't have to control my own muscle and think about controlling my heart, and glands, usually unaware of these actions, divided into two divisions, sympathetic and parasympathetic. Okay. And I think we discussed them earlier. Let's see. Yeah, so parasympathetic, think of calming, and sympathetic's rousing. Maybe think of some word phrases to remember that, like PC, politically correct. Okay. So let me hurry up and get back to where we were. Okay. So again, parasympathetic is more calming and sympathetic is more arousing. Now this is just looking at cranial and spinal nerves. We're not going to memorize all these. And it's kind of interesting views of where the eyes are. Here we like someone laying back on a pillow with their neck arched in A, but it does show you where the frontal lobe is and where your eyes are. Okay. And then it does show you that in relation to the cerebellum, that's very far away from where your frontal lobe is and where the medulla is, which would be the before the oblongata. Okay, and then and B is a cross section of like your spine. Okay, showing your spinal cord in the middle. Okay, and then it's surrounded by the vertebra. Okay, which is supposed to be protecting your spine. But sometimes the spacing gets small and then you get pinched nerves. Okay. Yeah, we won't worry about C. Okay, and then this is talking about the autonomic system, okay, which is not under your consciousness usually, but they're showing you the central nervous system, which includes the brain and spinal column in the middle, okay, and then talks about parasympathetic, which is supposed to be calming, and sympathetic is supposed to be more arousing. And so then they give you examples here of how parasympathetic is calming, constricts your pupils. So here, they're more closed. Usually like when you go from where it's dark to sunny, that protects your eyes. And then it talks about inhibiting heart, not making it pump as much, constricting the bronchi, stimulating stomach motility and secretion, and intestinal motility, and contracts the bladders. So they know many things that it can hurt here. You don't have to memorize all this. Sympathetic, which is supposed to be arousing, dilates the pupils, your eyes are wide open. But then it has some opposite effects that are shown over there. Like here it says accelerates heart. Okay, that makes you're excited about something. Maybe you're doing flight versus fright response. Okay, dilates the bronchi. Okay, so you're going to be breathing faster. Maybe you're digesting faster, maybe too fast. But at the same time, it's inhibiting intestinal motility, which is a little bit confusing, and then relaxes the bladder. So, okay, so it does mention various parts there and what they do, but we're not going to memorize that. But it's just some examples there that are very interesting. But I think the easiest one to remember is the eyes. PC being calming, constricts the pupils where you're like frightened by something, your eyes might become wide and explains your higher height rate and breathing rate with the sympathetic division. Drug abuse and how it's related to the brain is mentioned here. Person takes a drug at a dose level that increases the potential for harmful effect. So the definition of drug abuse, you overdo it. Okay, you're going beyond the prescription. Addiction is, unfortunately, your body's gotten used to a certain amount, and now they have to take more and more, and they can't get away from not taking it as needed anymore. Withdrawal is effects you have when someone suddenly stops taking it. Alcohol, drugs, and tobacco can adversely affect a developing embryo, fetus, or newborn. Studies have found that out. And sort of like secondhand smoke affects people that don't smoke. So they have looked at studies where they looked at drug use, and particularly they compared the brain activity before and after the use of cocaine. So we see that the highest brain activity is like blue to purple, and the brain is more active with the cocaine than without it. However, sometimes they're overactive and they do dangerous things. Okay, and then there are unfortunately neurodegenerative disorders that affect the brain, including Alzheimer's disease. Okay, and if we look at the two pictures of the brain here under Alzheimer's heading, the brain looks like it has shrunk, literally has shrunk. Okay. Okay, so there's shrinking of the cerebral cortex, but the ventricles, which is a little confusing because we did have ventricles in the heart, these are different. These openings have gotten bigger, and the hippocampus has also shrunk. Okay. So again, we have shrinking of the brain overall, but the ventricles or openings have gotten bigger. And then there's Parkinson's disease. You may be familiar with Michael J. Fox. Unfortunately, that is a neurodegenerative disorder as well. They have certain characteristics which include a hunched walk. They usually shuffle their feet. There's many other problems. It does affect speech as well. But if you're interested in other characteristics of Parkinson's patients, you can look at it here. Okay, I can try to go to that link here. I don't know if it will share the screen or not. Probably not. I'll probably have to reshare the screen. It says I'm sharing the screen, but I'm not really convinced. Let's see. Hopefully I am. But if not, we'll see. So it does, if you can't see it, you can look at the website yourself. And it does talk about, it's an age-related degenerative brain condition. However, Michael J. Fox did have it early on in his career. You may know he's an actor. But it's best known for causing slow movement, tremors, balance problems, and more. Some cases are unknown reasons, but some may be inherited. It's not curable at this time. That's why people want to do stem cell research to see if they can help. So it's a condition where part of your brain deteriorates, causes more severe symptoms over time. It does affect muscle control, balance, and movement, but it can have other effects on your senses, thinking ability, and mental health, and more. And there are pictures showing you that it can cause loss of sense of smell. Of course, this would not be like a cold. Drooling, constipation, gastrointestinal problems, sleep problems such as restless leg syndrome, mask-like facial expression. I guess it almost be like Botox. Slowed movements, and this is under the motor-related symptoms. Tremor while muscles are at rest. Rigidity or stiffness of muscles. Blinking less often than usual. Trouble swallowing, and they can actually aspirate. An unstable posture or walking gait. So usually they need a walker or something for the help. Unfortunately, there's no cure at this time. If we were able to do cell division in our brain, that would help, but we don't. But thanks for listening. Have a good day.